Hey folks, welcome. Come on in. You might have heard the expression. Sometimes people say, if you want to predict the future, then you should create it. And that's kind of what it feels like for me standing up here every day. That's what I'm doing. I'm trying to create the future that I'd like to see, where people are scientifically literate enough to understand what science is good for and what it's not good for, what its power is and what its limitation is. So that's what I'm hoping to do, helping you people understand genetics and genomics and science in general, and help you develop some skills so that I'll have a better life for the next 25 years or whatever I've got left. If you also would like that kind of future, then you can help me. You can do this. You can give me some really good, usable, actionable feedback on this course and how it has gone for you, so that I can make it better, so that we can all kind of have a better future. As I say that, can you get on this? Like, is this working? Try to, there's 182 people signed, but that hasn't moved in 10 minutes, so I'm worried that the site's not working. I've got a question coming for you in a couple of minutes. Has someone tried and not been able to get on, or you are on now, but it hasn't moved? Oh, cool. All right, so it's just not keeping track of the people. All right, so you're going to have an opportunity near the end of term, which I guess this is the end of term, but closer to the end of term, you'll have an opportunity to provide feedback in class if you want to do that. Otherwise, you can do this, of course, online anytime you like. This particular class finishes off cycle 11. Dennis is getting very lonely. He wants to come back. So Dennis is going to be here on Wednesday and Thursday of this week. And then we'll have a kind of tag team joint class on Monday, Tuesday, and that'll be it. That'll be the end of your careers as first-year bio students at Western. Because no one fails this course, really. Well, some people withdraw, some people decide on a different career path, but of the people who write the final exam in this course, 3 or 4 of them will fail. Just because they've got other stuff going on in their lives and they just can't focus or whatever. But for the most part, you will all be back again in second year in September. But September is forever away, right? Like, the best part of being a university student is the summer is 4 months long. Like, that is amazing. All right, so lots to talk about today. Hopefully we'll get to the part about Dennis's mother being a fish. But I've showed you this slide a couple of times. You understand that sequencing is becoming very much cheaper, and that one of the things we can do if sequencing is cheap is we can look at what is the smallest genome that would support life. And this is the JCV version 1. Where we were trying to figure out what is the fewest number of genes that an organism might need so that we can create them ourselves, so that we can create life ourselves if we can sort this out. So in your lifetime, for sure, you are going to need to deal with this. Scientists in a lab somewhere creating life forms, custom made. You'll have to figure out whether that's a good idea or not, or how to regulate that. how to ensure that it's not going to be a problem. We got into a little bit into sequencing. We talked about that all sequencing is really synthesis. We sequence DNA by synthesizing the complementary strand. We sequence DNA by synthesizing the complementary strand, and we get data like this. Masses and masses of A's and T's and G's and C's. We have to try to make some sense of it. So here comes the poll. If you wanted to know the sequence of the human genome, how many chromosomes do you have to how many chromosomes do you need to sequence? Just from your own mind. Don't talk to your friends. If you're not on count two, shame on you. But if you're not on count two, then just write your answer down. Commit. How many different chromosomes? Listen, no, stop it. The first answers are wrong. Think slow. You are experienced students. Think slow. Think slow. How many chromosomes do you have to sequence if you want to know the human genome? Think slow. Okay, nothing much is changing. I might have to take back that comment I made about how many people might not pass the course. So we're fighting over 23 and 24. And 24 is gaining the edge here. Of course, there are 22 autosomes, and an X and a Y. 22 autosomes, an X and a Y. You need to sequence all of those in order to know the human genome. So the B's are right. 24 chromosomes. That's the answer you arrive at by thinking slowly. Jumping on 23 is the answer for thinking fast. So do your best on the test. I won't have any more opportunity to tell you this, but on the test, do your best to think slowly rather rapidly. Okay, so when you're faced with this kind of data, like where are the genes? Where is anything? Where are genes, protein coding genes, these ORFs, or where are the tRNA genes? Like how do you find any of this? And we mentioned one of the reasons why sequencing has become cheaper is that computing power has become cheaper. So computers do this process, this next step, called annotation. If someone says to you, oh, I am annotating a genome, what they mean is they're finding the genes and the sequences and the centromeres, and they're finding all the parts and labeling them. And of course, we have computer software to do this. Computer software scanning these sequences to find where the genes might be, to find the ORFs, as Dennis mentioned a couple of months ago. And I hope you can realize that in this stretch of double-stranded DNA, there are 6 possible frames, 6 possible places that the gene could be coded. 6 possible places that the gene could be coded, as shown by these 6 possible reading frames. How would we know which of these frames is used by a gene? What other information might be useful? What other information might be useful to help us figure out which frame is being used? Yeah? If we can find a start codon and a stop codon that's in frame, that's about the right distance apart, then that's a good indication. What else? Yes? A little harder? That would indicate the... So maybe we can find a kind of a Tata box or an AT-rich region where a polymerase might bind. Maybe we can find a sequence that looks like the promoter. Exactly. So we need all of these kinds of signals. We're looking for all of these kinds of signals to help us figure out where the genes are. So here's a sequence like that. And we can see up here at the top, here's a start codon. And somewhere down near the bottom, here's a stop codon. And here is, at the very, very bottom, a poly A clipping signal. This is actually a cDNA, the sequence of a cDNA gene, so you should not be surprised to find a bunch of A's here in the cDNA. But this is the idea. This is real DNA sequence of a gene. And we think it's a gene because it has all the parts of a gene that we're expecting. But another way to be sure this is a gene is to compare it with other sequences in a database. Compare it to other sequences that other people have already found. And if you do that with this gene, you find out, oh, this sequence is ubiquitin. This sequence was published in this journal. It's a sequence from a wild sheep in China. This gene turns out to be, very likely a functional gene because it has the same sequence as ubiquitin. So another way to be confident that an ORF is actually used as a gene is to compare it to other sequences using, for instance, this technology called BLAST. Dennis is going to invite you to work with BLAST, maybe over the weekend. I'm not sure of his plans, but that'll come up soon. Okay, so computer searches, computer comparisons, all very useful. And when we do that for E. coli, you've seen this picture before, about 5,000 base pairs, about 5,000 genes. And now, I think maybe it makes better sense for you that some of the genes of E. coli are coded on one strand, the other genes are coded on the other strand. So these blue genes are coded on the top, the yellowish genes are coded on the bottom strand. We tried to make this point before. But notice how tightly packed the genes are. There's very little space in a prokaryotic genome. There's tremendous selection pressure for genomes to be small in prokaryotes. So there's very little wasted space here, very little real estate that's not used for a gene. Now I want to just swing back now to the synthetic life story, because that's also prokaryotic. And look, now we're at JCVI 3, version 3, an organism with an entirely synthetic genome. This genome was created by humans sitting at a computer typing out a sequence, manufactured entirely artificially, and introduced into an empty cell, and it worked. Under 500 genes. So the minimal genome we now know is something under 500. Even though there's 150 genes here that we don't know what they do. We know that if those genes are mutated, the cell dies, but we don't know why yet. And look at this. This genome, this man-made genome, is the smallest one ever. Smallest genome known of any organism in the universe is this man-made genome. But remember a few days ago when we were wondering about this, how would you figure out which genes were essential? And we thought, oh, if you just compare a bunch of genomes and see which genes are common, the genes that are common to all life, those must be the essential ones. Well, it turns out that's not enough. Those genes that we identified by comparative genomics, all of those still isn't enough. This organism here, this man-made organism, has twice as many genes as it would have predicted to need. So hopefully you remember this. You were first-year students when Volume 3 came out with the smallest genome ever. This table we've looked at a little bit already. I just want to point out again the density. Protein coding genes per megabase. Look at E. coli, almost 1,000. And no one's ever going to ask you for these numbers, but just note that E. coli is very dense. Compared to you, you've only got 7 genes per megabase. 7 genes per megabase. And look, that's not because your genes are a lot bigger. Your genes are somewhat bigger than E. coli. Look, your average protein is 400 amino acids. E. coli is 300. So your genes are a little bit longer, but not that much. Your gene density is very, very much lower than prokaryotes. In the last term, I showed you a graph indicating what the various sequences were in your genome. Now your understanding is much more sophisticated, and I want to show you a more sophisticated version of this. And look, that big yellow slice, half the frigging pie, defective mobile elements. Mobile elements that have infected your genome over evolutionary time and no longer move. Garbage. It's a junkyard. Look at the next one over. That lime green slice. Defective viruses. Junkyard. They don't even function anymore. So that's 45% plus another 10%, 55% of your genome is just a junkyard. DNA that doesn't even get expressed. Look at here, protein coding genes, 1%. And we can find another 1% that is essential but not protein coding. That's it, 2%. A few numps. Look at the introns. A quarter of your sequence is intron. And this is an important idea because I think if I asked you to just draw me a gene with some introns and exons in it, I think your mind would draw the introns small and the exons big. Your mind thinks exons are more important, and therefore they must be bigger. No. Not at all. Look at this picture. These are your genes. The bright, dark pink parts are introns. The dark pink parts are the introns. The light pink parts are the exons. Your genes are by far mostly intron. Mostly intron. And that might be the opposite of what the picture you had in your mind. Okay, so look, your genes are very much more spaced out than prokaryotes, and because your genes have introns that can be spliced or not, then as this picture shows, you can make alternative versions of a protein by alternative splicing. Okay. All right, this is game day, so I want you to think a little bit about this next one. I'd like the men in the room to think about this top one. If I change that one base pair, could that change the coding region of more than one gene? And the women, can you think of the bottom one? Is it possible for gene A to be in the intron of gene B? Could one gene be inside the intron of another? So think about that for a moment. And please don't just sit there and wait for the answer. And please don't fall into the trap of the Google effect, thinking, oh, I can just look this up sometime. Work on it. Sort it out. Can genes overlap in these ways? I'll give you a couple minutes. Okay, let's take a shot at this. Gentlemen, could changing one base pair affect the coding region of more than one gene? Anybody take a shot at that? Yes or no? Yes or no? Think more than one gene? Lots of really good words. So if there's a base pair right here, gene A, this base pair affects gene A, but then what's the splicing part?
So I'm saying if you, for example, were to move your hand a little bit to the left or right, yeah, it would start there. When the gene is a different start at that point, it can
So there's one gene that starts here and one that starts here.
And they'll include different parts of the
Are they in the same frame? So two genes coded in different frames. Maybe, but that's a good question. Is that even possible? For two genes to be coded on the same strand but in different frames? Top question, yes or no?
The genes are coded on both strands of the DNA. So
Opposite strands. So if genes are on opposite strands, then a single base pair could affect both of them. They could be coded, one on the top, one on the bottom. So one way for genes to overlap is to be coded on opposite strands. And the first answer, genes can overlap on the same strand. That's just going to be really, really rare. It's going to be really hard for a given sequence to make sense for two separate genes in two separate frames. But viruses do that a little bit. Viruses are under such selection pressure to have such small genomes that they do overlap their genes a little bit. So what I'm inviting you to do is go back to your notes where you draw those pictures of genes, and then sit down and actually picture and convince yourself how those genes could overlap. Get the transcription and translation signals right, and see how they could overlap. Okay, ladies, one gene inside the intron of another? Like, is that possible? Could one gene be inside the intron of another? Possible? Not possible? Possible? Could one gene be in the intron of another? Yes?
So we said yes because we thought about alternative splicing, and the intron is so big. So you can have a start and stop codon for gene A inside the intron, and it won't affect gene B because, say, in like your liver cell, the splicing sequence
Okay.
is at the end.
Perfect. Okay, so stop there. Whenever your mind sees the word intron, the next thought that comes into your head is splicing. This has nothing to do with splicing. You're right, but for the wrong reason. This question has nothing to do with splicing. Splicing happens at the level of RNA. We're talking about genes being coded in DNA. So yes, one gene can be in the intron of another. Imagine gene A is the big one. It gets expressed in liver. Gene B is the little one that only gets expressed in skin. No problem at all. So long as the little gene is on the opposite strand. Again, coded on the opposite strand, there's no reason why you couldn't have one gene in the intron of another. So those are good diagnostic questions that you should be sure you understand and quiz your friends about. Once you think you've found a gene, then you can try to understand what its function is by selectively knocking it out and seeing what happens. And you already know about mutations and RNA interference as possible ways to knock genes out. But what we have not talked about yet is CRISPR. When you started high school, people started to talk a little bit about CRISPR. And now that you're in first year, you're starting university, this technology is already revolutionizing biology. We could do a whole course about CRISPR. We could certainly do a whole lecture about CRISPR. We've just run out of time. So I'm going to give it only 4 minutes, enough to inspire you to do some digging on your own. It's all over YouTube. It's all over everywhere. But a really important point I want to make is that this technology that is revolutionizing biology right now came out of nowhere. It came out of what we call curiosity research. It came out of the research of people who were just wondering what's going on. It's a bacterial system that bacteria use as a kind of immunity to viral infection. It's a kind of molecular immune system that bacteria use to degrade the chromosomes of infecting viruses. The people who were studying this were just curious about how bacteria protect themselves from viruses. They had no idea that this would be useful at all. And the point I'm trying to make to you is that I want to live in a country in the future that does useless research. It is essential to do useless research, to investigate problems that seem to be have no function at all. Because every once in a while we stumble onto this. We stumble onto something that changes the world. We would never have found CRISPR if we were looking for it. So it's very important for you as scientists, for you as member of society, to support curiosity-based research that appears useless. This is one thing that can happen. So look at CRISPR. CRISPR essentially is a nuclease. It makes double-stranded cuts. This enzyme called Cas9 is a nuclease that makes double-stranded cuts. That's what the scissors are doing. But look at the green part. The green is single-stranded RNA that we call a guide RNA. So I think your mind has already put this together. Holy crap. We can send a nuclease to cut a chromosome anywhere we want. Anywhere we want. All we have to do is we have to know the sequence where we want the cut to be. We make a guide RNA that is complementary to that sequence, and the CRISPR-Cas9 system will make double-stranded cuts precisely at that location. Once a cut is made, you know what happens. Non-homologous end joining will try to repair that double-stranded break, and an indel mutation will occur. So this is a way to strategically, surgically create mutations exactly where we want them. Even better is another kind of repair called HDR, homology-directed repair. Homology-directed repair. And if you look on the diagram, you'll see, in this case, we offer some donor DNA that is homologous to either side of the break, and we can end up with new DNA inserted into that break. This, my friends, is genome editing, very precise genome editing. So not only can we sit at a computer and type out a genome, we can edit that genome pretty much at will, pretty much any way we want. So instead of doing all that transgenic stuff, instead of putting genes into organisms like we've been trying to do for years, we could instead edit the genes that are already there. They would still be genetically modified, but they would be genetically edited. They wouldn't have any extra DNA in there, just editing. This has already been done on human embryos. Not human embryos that came to term, but people are already messing around with editing human genome. Oh, here's an embryo from the IVF clinic at University Hospital, but we know this embryo suffers from cystic fibrosis. So we can shoot Cas9 in there and edit that CFTR gene and cure that disorder in this embryo before it gets implanted. We're not quite there yet, but we're not very far. Again, your lifetime, you are going to have to deal with this. Editing human genomes is going to be a thing. We have to decide whether we're going to do that or not. So one way to figure out if a sequence really is a gene is to mutate it and see what happens. Another way to find out if sequences are genes is to see if they're expressed, to see if we can find mRNA. And this is a technique called microarray, and just watch this, follow me. This is is a solid surface. The black squiggles are exome. Exome. Parts of your genome that are actually translated. That's called your exome. So we have this plate with the human exome on here. And then we take the mRNA from some tissue, reverse transcribe it into DNA, tag it with a colored fluorescent tag, and then ask which of the original mRNAs here made cDNAs that are complementary to which of these exomes. So it's a way to discover which genes in this tissue, which genes in the blue tissue are being expressed at any given time. Which genes are creating an mRNA at any one time. So this could be a control tissue and a tumor, or an adult and a baby. It could be anything. These two different samples. And then we read this again with a computer, and we can figure out which genes are being expressed at any given time. So this is microarray. A field called transcriptomics. The transcriptome is that collection of all the mRNA produced in a given tissue at a given time. The transcriptome. But if you go to work in a lab this summer, some of the work might be done on these microarray chips, but it probably will also be done using RNA-seq. And that just means, watch this, once you've got cDNA made from mRNA, you can just sequence it. Sequencing is so cheap, you can just take all the mRNA from a tissue, reverse transcribe it, and then sequence all those pieces. RNA-seq. So both of these are powerful techniques in transcriptomics. And you will not then be surprised to find that the next stage is proteomics. The entire suite of proteins produced by an organism at any one time is the proteome. The entire collection of small molecules, chemicals in a cell, is the metabolome. You can see that we are gradually realizing that nature is not a collection of pieces. Nature is not a collection of pieces that can be separated and studied individually to give us very much knowledge. Nature is a network. Networks within networks. I'll show you this again in a few minutes. But this one protein, beta-catenin, at the center here, the red one, interacts with this whole network. And I'm sure many, many more than this. So please, if you are working in research and you think, oh, I'm studying this protein. No, you're not. You're studying this component of a network. Keep that in mind. Everything's connected to everything. We can't, if you take a protein out of its context and study it, you're not really learning about that protein. There's a huge part that you're missing. So just keep this in mind as you develop your careers. Everything's connected. This is a field of biology now that's called systems biology. If this makes sense to your mind, if this more holistic approach makes sense to your mind, then you are a systems biologist. And you could pursue that. Because look, when you ask a cell, what are your proteins doing? The cell says, oh, 10% are involved in communication. 10% involved with communication with other cells. Because I'm just one part of a network, I have to keep in touch with other cells. Oh, look, here's 5% response to stimulus and the environment. So now we're up to 16%. Transport, moving stuff across my membrane. So almost a quarter of the genes in a cell mediate this network, mediate these connections with each other and with the environment. So if you take nothing else away from this course, please take that idea, that it's all connected. You can't, I mean, Western science for hundreds of years has tried to think the world apart. What is the difference between eukaryotes and prokaryotes? And what is the difference between animals and plants? We've had so much emphasis on how they're different that we tend to forget that they're pretty much the same. Their basic biochemistry is the same. Okay, just stop for a moment. We're going to change gears a little bit and think about how long ago was it that Dennis Maxwell's mother was a fish? There was a time, right? If you take Dennis's lineage and go back in evolutionary time, and if you take fish lineage and go back in time, there was an organism that is the common ancestor of Dennis and pufferfish. There had to be an organism that is the common ancestor of Dennis and pufferfish. It turns out it's 300 million years. I don't know if that sounds like a long time or a short time. 300 million years since Dennis's mother was a fish. Look, by comparative genomics, that's what we're talking about now. If you compare Dennis's genome to a fish genome, you can work all the way back to the common ancestor and say, oh, that critter had 12 chromosomes. We can recreate the karyotype of an organism that's been dead for 300 million years. The next slide's a little bit frightening. Just start at the top. Start at the top. That's what we think the karyotype looked like of Dennis's fish mother. But look what's happened since that time. In the lineage of Dennis, mobile elements went crazy. And we've seen that. Half of your genome is mobile elements, dead ones. But in 300 million years, mobile elements went crazy in the mammalian cell lineage. And then they broke and they fused together and the pieces moved around to give the modern human karyotype. But look at pufferfish. No crazy expansion of mobile elements. Pufferfish have almost no junk. Almost no mobile elements. But what they did is duplicate their entire genome. Whole genome duplication in the lineage that gave rise to bony fish. So by comparing the modern karyotypes of these two organisms, look at the dark blue pieces. Mostly together, like they're in chunks that are mostly still intact. So by studying the distribution of these chunks, how'd that get up there? By studying the distribution of these chunks, we can recreate the karyotype of these ancestors. Okay, so that's another sort of whole area of research, what we'll call comparative genomics. How can we reconstruct evolutionary phylogenies by comparing whole genomes, not just single genes? And if this is intriguing to you at all, then on the beach this summer, read this book. In particular, if your interest is in medicine, read this book. Your Inner Fish. Because at one time your ancestors crawled out of the ocean and gave rise to land organisms. At one time you were a fish, and there's still a lot of fishiness in your body. That is fascinating. Okay, so my final slide, my final message to you comes back to this idea of networks. So think about this for a moment. There's a mutation in a gene. It then changes the quality of the gene product, changes the way that product operates in the network, which then changes the cell physiology, changing organ physiology, changing the behavior of the person, changes how they act in their social relationships, which in turn affects the whole community. A single base pair change resulting in, well, I was going to say Huntington's, but that's many base pairs, but whatever. A change in genes can give rise to changes at the community level, for sure. But think of it the other way. The way people treat each other certainly is perceived by the brain, changes the neuron connections, changes the hormones that are produced, changes the networks, changes the epigenetic regulation of genes. Okay, again, it's just all one big network. So as you go into second year, people are going to want you to think like genetics, like cell biology, like organic chemistry. They're going to want you to divide the world all up. But try not to do that. Try to see connections between genetics and cell biology and biochemistry and organic chemistry. See it as one whole package, not a whole bunch of separate little things to try to remember. I think that's it. Okay, so Dennis will be here on Wednesday, and I'll see you around.